Tonight's subject is Neanderthal DNA, and this presentation was inspired by two individuals. I mentioned Neanderthal DNA in my new book, and one of the proofreaders said to me, "Are you sure?" And I realized, I guess it's not common knowledge that we have Neanderthal DNA, so I probably should fill people in on that. And the other one was, I posted something about having Neanderthal DNA online, and my mom wrote on Facebook, "That's from your father's side of the family." And thus I invented Grandpa Thag here, Neanderthals from my family tree. So we can start here in 1856 with the discovery of this skull cap in Neander Valley, which is what Neanderthal means in German. This was basically the first time that people recognized and understood that there was something unusual about this skull. And as you can see, this is slightly before Origin of Species had been published in 1859. So this was still pretty early. There had been two other Neanderthal remains discovered prior to this, one in Belgium and one in Gibraltar. But this one was sort of the first one that sort of people recognized this is different. And a little timeline then, 1864, we have the name Homo neanderthalensis, man of Neander Valley, and William King there proposed that it was a separate species. 1886 then, two skeletons from Spy, Belgium demonstrated, and this was important, demonstrated that these skeletal features were consistent and recognizable. So prior to this, people had speculated, well, maybe there's something wrong with these guys. Maybe they've got some sort of vitamin deficiency. Maybe they're just, birth defects or something. And with the discovery of these skeletons from Spy, people began to realize, no, these weird features on these skeletons are consistent. We see them in multiple skeletons, which is not what we would expect from birth defects or other deformities. 1912, then, we have Boole publishing a very famous and very ape-like reconstruction of La Chapelle-Usson, which is this skull here. We'll come back to this here in a minute. This is sort of the beginning of your classic ape-man characterization. They wear skins and they carry clubs and they're very stooped over and extremely hairy, and they look like apes basically. 53 to 57, the 10 Neanderthal skeletons were excavated in Shanidar in Iraq. And one of those was believed to be an actual burial. So this is one of the times where we have something where the anthropologists have suggested that this guy was put there on purpose. And the reason that they said that was the soil around the body was found to have what they contended was an unusual concentration of pollen, which they thought was from flowers being laid on the body when it was put to rest. Later, anthropologists have argued that it wasn't really that unusual and there wasn't anything special about it, so there's still some argument about exactly what that's all about. So in 1997, then, we have the first Neanderthal DNA sequences, and we'll get to what that all means here in a moment. Wanted to show you some of the fossils. These are mostly casts of the fossils. This is the very first Neanderthal fossil discovered. It is a child, maybe 3 or 4 years old, and originally discovered in 1829 in Belgium. So the fact that we are finding juvenile Neanderthals that have these recognizable features indicates again that these guys are different. They're really different. They're not just abnormal modern people. They are unusual, strange, different kind of people altogether. Then we have Sissaco Pastore, found in Italy in 1929, near Rome. Nice skull there. They are found in Croatia here. This was found in 1899. Has that very distinctive brow. And then in 38, Tash, found in Uzbekistan. Uzbekistan, if you have no idea what that blob is, that's the map of Uzbekistan. We are Americans, I'm afraid, so we don't recognize Uzbekistan, but that would be Central Asia, be straight north of Afghanistan. And so you can see then there's a wide geographic range for the Neanderthals. They go from the sort of the farthest westernmost points of Europe way over there into distant Siberia. The blue squares here, the blue circles represent what are called pre-Neanderthals. There's some disagreement about exactly what they are, but they may well be sort of early versions of the Neanderthals that haven't yet developed the full sort of classic shape of the Neanderthal skull, the classic features of the Neanderthal. All right, and we will get back to Oaza later. That guy right here in Romania, he's kind of important. He's not exactly a Neanderthal, but he is pretty cool. So what were they like? This is a famous mural from the American Museum of Natural History showing one of these early depictions of a Neanderthal wearing their skins and having their spears and big hairy things. This is actually quite a bit better than the earliest Neanderthals, which were basically hairy all over and look like gorilla or guys in gorilla suits. These guys are at least, they're mostly human. They just look like they need a haircut. But the face is still quite different. And frankly, that's not unjustified. So the basic features of the Neanderthal, this is La Chapelle, as I said, low forehead here. You have a rather pronounced brow ridge, which is something that we definitely lack. There is the eye sockets tend to be round. The mid-face here protrudes. I've been told it's as if someone grabbed your nose and just pulled it straight out. That's the classic Neanderthal face. The back of the skull here, this is the occipital bone here, and it has basically an enlargement here called the bun, the occipital bun. The brain case is large. It's actually on average larger than Homo sapiens, so they have a bigger brain than we do, even though it's a little bit squished and flattened this way. And then the mouth apparently is large enough that there's a noticeable gap behind the third molar. So apparently there was enough room in a Neanderthal mouth for the wisdom teeth, which generally speaking sort of crowd into our mouths and get pulled out by dentists afraid of infection. So those are the basic Neanderthal features. More modern reconstruction here at the Neanderthal Museum shows mostly a light-skinned individual. The face is definitely different, but not nearly gorilla or chimpanzee different. You would recognize them if you saw them. You would say that's a funny-looking person, but not so much that you would think it was something completely different. They still have them dressed in skins, though, but we don't really know how they dressed. All right, so what we really want to talk about tonight is the DNA. What is this whole business about Neanderthal DNA? As I look out at the crowd here, we all have it, everyone here. And so how do we know this? Well, very simply, we'll start off with just a very brief reminder of DNA. DNA comes in physical bits called chromosomes, which are on the far right over there. they're really, really, really long strings. They're like beads on a string. And the beads come in 4 flavors, A, G, C, and T, which you can just sort of, because they're linear strings, you can just sort of represent the whole thing as a row of letters. The human genome contains about 3.3 billion of these letters, which is pretty staggering. If you printed that out, it would take many, many books and lots of paper and be very wasteful. No one would want to print out the human genome. That would be inefficient. The various chromosomes we have, 22 what we call autosomes, and then the sex chromosomes, which determine our sex. So if you have a Y, you're a boy. If you have 2 X's, you're a girl. Pretty simple stuff. This is an actual karyotype that I got from the CDC. Karyotypes being actual microscopic images of the chromosomes when they are all condensed up and coiled, which is when you can actually see them under the microscope. And you can see there's some patterns here that look like banding, and indeed that is exactly what it is. There are parts of the chromosomes that coil more densely than other parts. That's very interesting. You might also notice here on the bottom right, seems to be mislabeled. Those are 2 X chromosomes rather than an X and a Y. But otherwise, that would be, if you could see them under the microscope, that would be what they would look like. So what have we learned from human genomes? So a genome is essentially all the DNA that a cell contains. So we are made up of cells, we are made up of trillions of cells, and each cell, most of our cells, contain essentially the full DNA that is needed to build us, which is why people are interested in adult stem cells, because you can still get all those chromosomes in there that make up a person. And so it'd be interesting to see if you could sort of deprogram the cell back to being something that hasn't turned into an organ, so that you can see if you can grow a new organ. Anyway, so it's all because those cells contain that DNA. Now, since the human genome was published back at the beginning of the millennium here, there has been an effort to sample more widely human variation in the 1,000 Genomes Project, which long since has passed 1,000 human genomes. There's now well over 2,500 human genome sequences, which, when you think about it, is pretty staggering. It took more than a decade to produce the first human genome, and now in the past 2 decades, we've produced over 1,000 of them. We have documented, scientists have documented almost 162 million variations between different individual people. So if I compare my DNA to your DNA, there would be differences, right? Some of us would be obviously different because some of us have 2 X chromosomes and I have a Y chromosome, so that would be obviously different. But there are other variations that give us hair color and eye color and height and weight, and there are different variations that contribute to those things. So what we found in modern genetics, the old idea of, if you have this gene, you have brown eyes. If you have this gene, you have green eyes, and so forth. That's seems to be very unusual when it comes to human traits. Actually, most traits there are multiple genes that contribute to them. Height, weight, all that stuff. So most of these variations that have been documented are what are called single nucleotide polymorphisms. There are a number of other things that are more substantive and larger, but most of them are very small. So an average genome, if you compare, say, my genome to the original first, what they call the reference genome sequence, I'm going to differ from that by about 4 to 5 million locations in my genome. And that sounds like a lot, but it's really 0.1%. So you sequence the genomes in this room, 99.9% of it is going to be exactly the same for all of us. It's those 5 million or so sites that are different and make us different. And then there's a much smaller number that are structural variants. And yeah, we differ very little. Now, single nucleotide polymorphism, that is basically where you have one of the beads on your string that is different from somebody else. That's all it means. So, and it's variable within people, right? So I compare my DNA to your DNA. I have a T at this place, and you have an A at that place. That's a single nucleotide polymorphism. And since we don't like to keep saying single nucleotide polymorphism, we just call them SNPs. It's much easier that way. So that's a SNP. And you know, mostly, that you know where the SNPs are because 99.9% of the rest of the DNA is the same. So you can line it up pretty good with really good confidence that you're looking at the same spot in the genome, and you have one nucleotide that I don't have. So there you go. And from those SNPs then, largely from the SNPs, we get all of the variety of people in the world today. Those SNPs seem to be correlated with every genetic feature that you can imagine in people. It is remarkable because there is such a diversity. Some people have, you know, a predisposition to, say, heart attack. Other people have predisposition to addictions. You have really tall people, really tall families with really tall people in them. And you have really short families with really short people in them. All of that stuff is SNPs. That's all, well, most of it is SNPs. There are some variations that are larger, but most of the stuff is SNPs. Now, what's really cool about this then, we can look at parts of the genome and we can compare them, and it sort of makes sense if you're going to compare two genomes. Genomes that are really, really similar are probably closely related. If you take all of those variable sites and you look at them, so for example, if you compared my genome to my parents' genome, you would see mostly an exact match. There might be one or two differences between me and my parents that I did not inherit that are just spontaneous new mutations, but most of the variations are going to be exactly the same. My cousins, I will be more different from my cousins. And then the farther back you go, you know, more distant cousins are even less similar and so forth. And you can then take all those similarities and you can map them out on a family tree, which is kind of cool. And this particular family tree is composed of DNA sequences from something called mitochondrial DNA. Mitochondrial DNA is a very small bit of DNA that happens to be inherited from your mothers. So mothers pass it to their children. Men do not pass theirs along most of the time. I think there's a very small number of known examples of paternal inheritance, but the vast majority of this mitochondrial DNA is only inherited from your mother, which makes it kind of convenient for sort of mapping things out. So here's a big family tree showing the mitochondrial DNA from a bunch of different modern humans, living humans. And you can see we have a big branch here that represents Africans and non-Africans in yellow. And then the blue branches here are the sub-Saharan Africans, which turns out to be really interesting. Those guys in sub-Saharan Africa are the most different from any other people in the world. So you take two random people in the world, compare their genomes, they will almost always be more similar than if you compared either one of them to somebody from sub-Saharan Africa. So these guys are the most different from everybody else. And this is why people talk about, evolutionary biologists talk about out of Africa when they talk about where humans come from, because the most different humans live in sub-Saharan Africa. They're tribesmen from sub-Saharan Africa. All right, well, that's all well and good, but what about the Neanderthals? Well, as I said, in '97, The first small sequence of Neanderthal DNA was published. It was from a piece of mitochondrial DNA, and it was pretty strikingly different from modern human DNA, which was really interesting. It was well outside the range of what we'd seen in modern humans. And since then, scientists have sequenced Neanderthal DNA from a range of Neanderthals pretty much across the entire geographic range. They haven't really gone very far south, but certainly east to west, they've gotten a huge sample of Neanderthal DNA. And initial efforts to do this were difficult. They were trying to basically make biochemical copies of the DNA before they sequenced it. Sequencing meaning, get the order of the beads on the string. And more recently, the newer sequencing technology, the stuff that lets us do the 1,000 Genome Project, that sequences the DNA directly. You don't have to make copies of it. You can just isolate the DNA from the bone, the ground up bone, and run it through your sequencer, and there you go. So that's what makes, I think, a lot more exciting results coming through the pipeline as we learn more about these guys. So here are a sample of Neanderthals from across their range, showing you how they relate to the living people. Remember, those bottom branches there are the sub-Saharan Africans for living people, and then you have all these Neanderthals. People ask me, how can you be sure that the Neanderthal DNA is really Neanderthal DNA and is not somehow messed up, right? If it's really however many thousand years old, and it's been sitting in the ground, come on. I mean, you put DNA into a fridge that's fresh from a bacterium or some living organism, it doesn't last that long. It goes bad pretty quickly. How do you know that that DNA from that Neanderthal is good and is really telling you something? Couple of ways to think about that. One of the ways to think about that is the consistency of the DNA. If we were looking at random errors in the mitochondrial DNA, I would expect them to be random, right? I would not expect them to show up at the same places and therefore be consistent, especially if I'm looking at Neanderthals from, say, Siberia versus Neanderthals from Spain. I would have no expectation that there would be any sort of common climate conditions that would cause those things to have exactly the same chemical degradation at exactly the same spots. So the fact that these Neanderthals here all group together just like the living people do, well, that tells me these are probably real differences because the Neanderthals are all very similar to one another. Another way that we can do this, and this has become more common as Neanderthal genomes have been sequenced, you can actually use the chemical damage on the DNA to show that it's really Neanderthal DNA, which is kind of cool. So what you find is the damage on the DNA usually happens very close to the ends. And what goes on when Neanderthal DNA is sequenced directly from the bone, the fragments are very small. So the human chromosome 1 is 250 million nucleotides long. It's gigantic. It's our largest chromosome. So what you get when you sequence a Neanderthal genome are pieces that are 50 nucleotides long. Really, really, really tiny. And the majority of the differences are near the ends, typically within 10 to 20 nucleotides of the ends. So that suggests that the stuff in the middle is not too bad, but the more important thing is you can recognize the old stuff, right? So how do you weed out modern contamination? What if by some accident they weren't careful enough in their clean room and a piece of dandruff got into the sample, the Neanderthal sample? Well, you would know it because it doesn't have that characteristic damage on the end of the sequences. Cool. So that's another way that you can sort of pin this down. And the third way they would do it, they would focus on locations in the human genome that are known to be variable in modern populations and compare those to Neanderthals. And in doing that, then, even though you have a pretty high error rate in your ancient sequences, by focusing on the places that are highly variable in the modern populations, you're looking at things that are very likely to be variable in Neanderthals as well. And so you can ask questions about how often do we match the Neanderthal sequence, how often do we not. And so your, say, 3 or 4% error rate in your ancient DNA becomes much less important, right? Because the differences among modern people are 0.1%. So 3% error rate is huge. It could be overwhelming the real biological signal. But that's why they look at these polymorphisms. The fourth way then is to just sequence the same piece of DNA 40 times to make sure you really got it right. And make sure that that sequence is really what's in the ancient sample. So there's various ways of dealing with this, all of which I find extremely compelling and persuasive. And I think, yes, that's real Neanderthal DNA. They really have it. It's available on the internet. You can download it and look at it for yourself. It's really cool. But we'll talk about that in a moment. What was really surprising, and this has turned out to be one of my favorite results of the past 20 years. Now we know about an entirely different group of people that go by the name Denisovans. They're known simply from tiny bone fragments and a couple of teeth from this cave in Siberia, the Denisova cave. We have no idea what they look like. We can see that the teeth they're different. Once you realize that the DNA is different, you can see that the teeth, yeah, they are kind of different from Neanderthal teeth and from modern human teeth. But we don't have the full body fossil yet to say what they would look like. But we have their DNA. So now paleontology is entering this whole crazy new realm where you can find new things that you never knew existed, even from the scrappiest, useless, most junk piece of fossil that no one would have thought was important before. Now they're in, masks and hairnets and gloves, sampling this stuff out of the ground to make sure that they don't contaminate it, because even the junkiest piece of junk might contain an amazing new discovery in the DNA. All right, so let's talk a little bit more about that. This guy here is the main man when it comes to Neanderthal DNA. This is Svante Pääbo. He is, well, I've followed his career for a very, very long time. In 1985, he extracted mummy DNA for the first time. He was a postdoctoral student at the time, and managed to show that Egyptian mummies did have what appeared to be DNA still in them, which was kind of cool. 12 years later then, we had the first Neanderthal DNA fragment, and 13 years after that, his lab produced the Neanderthal genome. And they got it, as I was saying, from these three basically scrap bones from Croatia, from a place called Vindia Cave. Okay, so what do we find when we get this DNA? Well, this is the main interesting result that everyone wants to know about. So what they did here, they compared, in addition to sequencing this Neanderthal genome, they sequenced a sample of modern human genomes with basically the same method. So they did a sub-Saharan African, an Asian individual, and Europeans. So I think they did 2 of each, actually. And so what they found was, when they compared the Neanderthal DNA to an African, and then compared it to a different African, so pairwise comparisons, they found that, well, the Neanderthals seemed to be roughly equally different from each. However, when they compared the Neanderthal DNA to a non-African and then to an African, they always found that the Neanderthal DNA matched the non-African better than the African, which was really odd. They thought, well, what is this? What is going on here? And then, of course, they tried that with non-Africans again and found essentially a random distribution there. There didn't seem to be any major bias where it was more similar to one non-African than another. But consistently across the board, no matter how many pairs of genomes they compared, the non-Africans always seemed to be more similar to the Neanderthal than the Africans. Now that's really strange because the mitochondrial DNA that they had studied showed that these guys were different and consistently so. And yet here, this seemed to be saying, actually, there is a significant reproducible similarity between Neanderthals and non-Africans, Eurasians, than with Neanderthals and sub-Saharan Africans. And how does that work? What's going on here? Well, some basic genetics here. Every time cells divide to produce sperm and egg cells, they undergo a process called crossing over. And crossing over, you take two chromosomes, we have the red one and the black one, and they literally form a little cross. And that's why it's called crossing over, right? And then at that juncture, they will literally be broken and stitched back together again. So that this piece here ends up on the other one. The red tip ends up on the black chromosome, and the black tip ends up on the red chromosome. So that if you were to actually look at your chromosomes, you got half of your physical chromosomes from your father and half from your mother. But if you compared your chromosomes to your grandparents, they would be like a quilt. So that chromosome 1, well, part of it might come from your maternal grandmother, and the other part might come from your maternal grandfather. So that each generation inherits this interesting stitched together piece of DNA. So after many generations then, if you start out with all these nice chromosomes, you literally end up with this quilt-looking thing. And generally speaking, the more generations you go, the more crossing over happens, the smaller the pieces get, right? So you can sort of estimate how many generations, assuming, of course, that crossing over has always happened at the same rate. You can sort of estimate how many generations back you would go to find when you shared an ancestor. So anybody have 23andMe or something like that? Those guys will tell you, oh, we found a close relative of you. He is probably your whatever cousin twice removed or something. And they're doing that because they can see, oh, there's a bunch of variable places in your genome that match exactly with this guy, and it's this long, and that is most likely because you share a great-grandfather or a great-grandmother or something like that. That's all coming from this sort of thing right here. Now, what makes this really interesting for our purposes is if you have something that's relatively homogeneous, where you have things that are very similar, and you throw in a chromosome that's very different, well, after many generations, you can still tell where that is, right? Because it's kind of different. And so with Neanderthals, that's what we think is happening. If you know what that looks like, then you should be able to recognize the spots in our own genome that look like Neanderthal DNA. And I am pleased to present to you my own Neanderthal DNA. There it is. Tiny little bits across the genome that reflect the likely places that my chromosomes have bits of Neanderthal DNA. This is data from 23andMe, in case you're wondering. So you sign up there, they'll send you a little box, you spit in a tube, you send it back, and then within the month they will have your data available. You can download the raw DNA sequence, which is pretty cool. or you can just look at their little charts that they generate, and they will tell you what kind of DNA you have. I am pleased to say I did inherit a Neanderthal gene that seems to have an important function. It's correlated with less back hair, so that's nice. Yeah, it's really interesting. You know, all of this Neanderthal DNA is present in these genomes, and yet, you know, knowing what they do and what it's for, there's only a handful of these genes, at least in the 23andMe sample, that they have some sort of correlation to actual function. There have been other studies that have shown more important functions, more interesting functions. But for me, I just have no back hair, which is kind of nice. So that's my Neanderthal DNA. There it is. All from my dad's side, as my mom says. Yeah, not how it works. Now, what's really interesting here, and this is another reason I think this Neanderthal DNA result is real and not just an artifact of errors. If you look across chromosome 1 here, and look at how frequently you find, you know, you go back to the 1000 Genome Project and you ask, okay, well, how often do I find what look like Neanderthal genes at different positions on chromosome 1? And you'll find here that it's not uniform across the chromosome. There are places on chromosome 1 where there's literally no Neanderthal DNA detected in modern populations at all. And then there are other places where it seems to be pretty frequent in modern populations. So that's really interesting, and that makes biological sense. If you're going to put two different genomes together, there are going to be places where the genes are diverged enough that they don't really work together. And so over time, those parts, people who inherit those chromosomes that don't work well together, they're going to be sort of weeded out of the population. So what you end up with is something like this, where you have places where there's a lot of Neanderthal inherited and places where there's not, or places where there's none. And cool, very neat, you map out my Neanderthal sequences. There's only a handful of them from chromosome 1, and they line up pretty well. I mean, it's not perfect, but they do happen to hit these places where we all have Neanderthal DNA. So I'm not unusual in that regard. All right, so a random Eurasian individual in our modern world has about 1 to 4% of their genomes that have been inherited from Neanderthals. So that's interesting. But what if we got everybody together and we went back and tried to stitch together a living Neanderthal genome? Could we do it? How much of the Neanderthal genome survives in modern humans? And you might guess already, not all of it, right? Because I already showed you chromosome 1 where there are patches where there's no Neanderthal DNA. So we already know there are places where we have missing DNA. And it turns out there's only about 20% of the Neanderthal genome still around in our modern chromosomes. So no, you could not get people, whatever, you know, biotechnology or whatever, and rebirth a new Neanderthal. That wouldn't work. Only 20% of it still exists. Now, there have been additional surprises, and that's what makes this cool, because, of course, having Neanderthal DNA means that at some point in my family tree, my ancestors married a Neanderthal. Or maybe didn't marry, but they had children with a Neanderthal somehow. And what makes that interesting, so I mentioned Oaza earlier, that Romanian fossil. That one is a modern Homo sapiens, but it had always been suspected to be something of a hybrid because it has some weird Neanderthal features. Not enough to say this is definitely a Neanderthal, but enough to say, wow, this guy's really weird. Sequence the genome, and sure enough, he's got really long stretches of Neanderthal DNA in his chromosomes, implying that he has perhaps a great-grandparent or a great-great-grandparent that was a full-blooded Neanderthal. So a very recent ancestor. And the most recent discovery, Denisova 11, that same cave where we found the Denisovan individuals, a tiny little bone fragment. So I mentioned earlier that you can get really great, amazing discoveries now from basically garbage bone that no paleontologist would care about. So this is a copy of a 3D scan of a bone fragment found in the Denisova cave. This tiny little nothing that would be unimportant. I can't even hold it. It would be unimportant to paleontologists in past generations. Turns out it is a first-generation hybrid between a Neanderthal and a Denisovan. The sequences from Neanderthal and Denisovans go all the way across the chromosomes evenly, indicating that this individual had one set of chromosomes from Neanderthal and one set of chromosomes from Denisovan. Even more interesting, there were a handful of very small segments that were totally Denisovan and not Neanderthal at all, implying that the Neanderthal parent had some Denisovan ancestry as well. Crazy stuff. And we know that's a Denisovan mother because the mitochondrial DNA, which is inherited from the mother, is Denisovan and not Neanderthal. So this tiny little bone fragment, which would be unimportant to generations past, now becomes a treasured discovery of science because, oh my goodness, it turned out to be this first generation. So we know these Neanderthals and Denisovans were interbreeding and having offspring. All right, so what does all of this mean then? Well, the basic model is something that looks like this, where we have the sort of the large lines here represent, as best as you can guess, populations that diverge. Think of it as a big giant family tree, right? And there is a large divergence here between Denisovans and Neanderthals. They seem to be roughly cousins. And then there's the divergence between Eurasians and sub-Saharan Africans. And this is more distant in the past, where we all sprang from the same original group. And then we have interbreeding between Denisovans and Neanderthals, as indicated by this little fossil. We have interbreeding between Neanderthals and Eurasians, but not sub-Saharan Africans. And then we also find evidence of interbreeding between Denisovans and Eurasians, where it looks like, especially people from Papua New Guinea, seem to have Denisovans in their ancestry as well. So there's a lot of intermingling going on between these ancient human individuals. Now, since we're here to hear about creation, what does this mean for all of this stuff? I mean, what do we do with this? My first inclination, my first thought on that matter is I don't think humans and animals are breeding together. I don't think that's really going to work. I don't think that makes sense in terms of our understanding of the image of God or our understanding of the human soul. Does an animal-human hybrid have a soul? Did Jesus die for that thing? How do we even approach those kinds of theological questions? That seems to me to be a prickly thorn bush that is just impenetrable. So I'm going to say, if they're interbreeding like this, they're all people. Seems the easiest thing. Now, what makes this interesting to me then, when you look at modern debates on the evolution or origin of human beings, you hear a lot of people talking about, well, you know, we don't have any Adam and Eve at the base of the human tree. You go back and look for Adam and Eve, you don't find Adam and Eve living together in one place in genetic data. The genetic data is way too complicated. All that switching and swapping of pieces of chromosomes, all that stuff suggests that there was never an Adam and Eve. But they're all looking for Adam and Eve where modern humans all begin diverging, right? That's where they go looking for this. And so I'm looking at this whole much bigger picture now and thinking to myself, they're looking in the wrong place. It's pretty simple. So if Denisovans and Neanderthals and modern Homo sapiens are all intermingling like this, I think we should really be looking for Adam and Eve down here. Much, much deeper in human history, genetically speaking. Now, what that means for, chronology, when you think these people lived, what they were like, those are interesting questions. I don't think that has anything to do with the image of God. The image of God is our position as the rulers of this creation that God has given to us. We have dominion. That is part of what the image of God is. The image of God is not, a forehead or a chin or a tooth or some physical feature that we possess. Every human being is the image of God, no matter what physical normalities or abnormalities they have. And so when I look at Neanderthals, I don't see something that is offensively unhuman. I simply see something that is different from the way we turned out in the modern world. So I don't think that threatens our understanding of the image of God or our theological place in the world, redemption. All of that is conserved simply by saying, no, I think Adam and Eve, humanity's origin begins way before you get Neanderthals, Denisovans, and modern Homo sapiens. Hey, thanks for watching my video lecture. If you liked that, do me a favor and hit the subscribe button. If you didn't like it, do me a favor and post your outrage on social media so that other people will see it and maybe they'll hit the subscribe button. Check us out on Facebook, Twitter. You can find our website, Core Academy, is at coursci.org. My own blog is toddcwood.blogspot.com. Thanks for watching.